Today we will discuss about Taenia species from Parasitology. It comes under Cestodes group. So here in the first slide you can see a comparison between Cestodes, Trematodes and Nematodes on the basis of shape, sexes, structure of head, alimentary canal and body cavity. So the most important part is shape. Stoats are tape-like, that's why commonly called as tapeworm, and trematodes are leaf-like, unsegmented, and nematodes are elongated, cylindrical, unsegmented. Second most important difference is shape of head end. So in case of cystodes, you can see suckers are present, often with hooks. In trematodes, suckers are present, but no hooks. And in nematodes, no suckers, no hooks, but head is well developed. So these are the main differences between all three groups. In the next slide you can see some important terms related to this lecture, this topic, like strobila, that is used for to describe the body, and scolax is for head. Proglottids are segments and carry eggs in case of tapeworm. Rostellum is to define a beak-like projection on head. Oncosphere is mainly to define embryo, and embryophore is enveloping membrane surrounding the oncosphere. Egg cell is outer membrane that enclose oncosphere with the embryophore. Cysticereosis is to define the resting stage of larva in intermediate host. And cysticercoid, a small bladder containing invaginated head proximally and a solid elongated portion that is related to cysticercus stage. Like Conidus, it's a larval stage in form of bladder worm. Hydrated cyst, that is used in case of echinococcus parasite, and that is also included in this lecture, echinococcus, and that is very important, hydrated cyst. Coracidium, it's a ciliated oncosphere of Diphyllobotherium, but that is not included in this lecture. So now we'll discuss about first parasite, Taenia. There are two different species, Taenia solium and Taenia saginata. The solium one is commonly called as pork tapeworm, and saginata is beef tapeworm. Mostly present in small intestine, and the infective forms called as Cysticercus cellulosae and Cysticercus bovis. They belongs to cystode and found throughout the world, and is the most common in countries where pork is eaten in case of Taenia solium. There are two different forms of Taenia solium. One is due to eating uncooked pork, that contain the cyst, and other is known as cysticercosis, is due to eating food or water contaminated with feces from someone infected by the adult worms. So in this diagram you can see pork tapeworm, Taenia solium. In the plate you can see clear diagram of pork tapeworms. There is no body cavity is present, and normally size is very large, 2 to 3 meter in length, but can become much longer, sometimes over 8 meter long. That is very big size. White in color and flatten into a ribbon like, tape like structure, tapeworms. And the anterior end is a knob like attachment organ called as head, also called as scolex, that is 1 mm in diameter. Now, life cycle of Taenia. The life cycle of Taenia solium is indirect. It passes through pigs or other animals as intermediate host into humans as definitive host. In humans, the infection can be relatively short or long lasting, and in the latter case, if reaching the brain, can last for life. For human, the eggs are released in the environment where they can await ingestion by another host in the secondary host. The eggs develop into oncospheres, which bore through the intestine wall and migrate to other parts of body where they form cysticerci. It's a form. The cysticerci they can survive for several years in animal. Here in the life cycle, you can see infective stage and diagnostic stage separately. In case of both Taenia solium and saginata, the diagnostic stage is eggs and the infective stage is oncosphere, develop into cysticerci and infect the humans. So it's a common life cycle for Taenia species. In the next slide, you can see separate life cycle of Taenia solium. It completes in two different stages. In the definitive host, humans are colonized by larval stage, the cysticercus, from undercooked pork or other meat. Each cysticercus is oval in shape, containing a inverted scolex, which everts once the microorganism is inside the small intestine. This process of evagination is stimulated by bile juice and digestive enzymes from the host body. Then the Taenia solium loads in the host upper intestine by using hooks and suckers. The four suckers are present and they enter into small intestinal mucosa. Then the scolex is fixed into the intestine by having the suckers and attached to the villi and hooks extended. After that, they grow in size by using host nutrients. Its strobilar length is as new proglottids are formed at the foot of the neck in 10 to 12 weeks after initial colonization. It is an adult worm. So the total time in the conversion to adult worm is 10 to 12 weeks. The exact lifespan of an adult worm is not determined. That is not known. However, evidence from an outbreak among some research in 1930s that indicate they can survive 2 to 5 years in human. It's on the basis of some US based research. So this is about the life cycle of Taenia solium. Some points also related with the second part that completes in intermediate host, that is a pig. These are the most common host who ingest such eggs in traces of human feces, mainly from vegetation contaminated with it, such as from water bearing traces of it. The embryonated egg enter intestine where they hatch into motile oncospheres. The embryonic and basement membrane are removed by the host digestive enzymes, mostly pepsin. Then the free oncospheres attach on the intestinal wall using their hooks. And with the help of digestive enzymes from the penetration glands, they penetrate into intestinal mucosa and enter into blood. Then move general circulatory system into various organs, and large numbers are cleared in the liver. They survive oncospheres and migrate to muscles as well as brain, liver, and other tissues, where they settle to form cyst called as cysticerchy. It's a very important stage. Single cysticercus is spherical and size is 1 to 2 centimeter in diameter. It contains protoscolex. Important point is that cysticerchy are usually found within 70 days and may continue to grow for a year. So humans are the also accidental secondary host when they are colonized by eggs, either by autocolonization or ingestion of contaminated food. So these are some important points related to life cycle. Now in the next slide you can see life cycle of another species, that is Taenia saginata, commonly called as beef tapeworm. The life cycle is almost similar in both cases. The main difference is you can see of the intermediate host. In the case of Taenia solium, that was pig, and in case of saginata, that is cattle. That's why called as beef tapeworm. And the second most important difference is the infective stage, name of infective stage in case of both life cycles. The cattle acquire the embryonated egg, the oncosphere, when they eat contaminated food, and then they reaches to duodenum, means in small intestine, and hatches there under the influence of gastric juices. Uh, if we start from the second step, you can see in the diagram. So similarly, like a previous life cycle, the infective and diagnostic stage is same. The embryonic membrane are removed after that stage and liberating free hexacanth larvae. With their hooks, they attach to intestinal wall similarly and penetrate into the mucosa and blood vessel. These all are the similar steps like Taenia solium. So this is about the life cycle. So in this diagram you can see clear structure of Taenia solium. The size is very large in case of this parasite, so you don't need a microscope to identify this parasite. Adult Taenia solium is a triploblastic accumulate having no body cavity, size 2 to 3 meter in length, and sometimes it can be more than 8 meters. So size is very big in case of this parasite. The scolex you can see in the diagram. The scolex bears 4 arranged sucker that around the rostrum. These are the organs of adhesive attachment to the intestinal wall. It's a very important part of body, and the rostrum is armed with 2 rows of hooks, spiny hooks. Its 22 to 32 rostellar hooks can be differentiated into short and long types based on the basis of their size. The adult has a flat ribbon-like body which is white and measures 2 to 3 meter. As I told you earlier, it's tiny long attachment, and the scolex contain duodenum wall suckers and a rostrum. You can see in the diagram. Here again you can see diagram from some different angles. You can see after a short neck is the elongated body, the strobila. The entire body is covered by a covering called tegument, and that act as absorptive layer consisting of a mat of specialized microvilli that called as microtrachs. The strobila is divided into segments called proglottids. And proglottids, here you can see uh, different segments of Taenia solium and Taenia saginata in both diagrams. The proglottids can be 800 to 900 in numbers, and body growth it starts from neck region. So the oldest proglottid are at the posterior end. Thus the 3 distinct proglottids are immature proglottids towards the neck, the means oldest one, and the mature proglottids is in the middle, and gravid proglottids at the posterior end. This is the three different terms used for proglottids. You can see the clear tapeworm like structure in this diagram that contain proglottids, collex, rostrum, neck, and clear head portion. The oldest gravid proglottids are full of fertilized eggs. Each fertilized egg is spherical and measure 35 to 42 micrometer in diameter. These are carried by these proglottids. Here you can see the real picture of a Taenia. You can see different proglottids like immature side and mature side. That is called as gravids. Here you can see the shape, size, and diagram of eggs present in stool microscopy, and in the second diagram, the green diagram, that is bile stained. They are helpful for diagnosis of this parasite. So the most important part for all parasites diagnosis is stool test that commonly include microbiological testing. After that, you can examine microscopically. The stool concentration aims to determine the amount of eggs. Specificity is extreme high for someone with training, but sensitivity is quite low because the high number of eggs is in a small intestine of sample. You can also use stool tapeworm antigen detection and stool PCR, serum antibody test for diagnosis of this parasite. Here you can see clear eggs of Taenia species. Here are different techniques used to diagnose the Taenia solium or Taenia species. Like in case of brain infection, you can use biopsy, X-ray, MRI, CT scan of brain calcification. You can also use ELISA and myelography in case of spinal infection. But you can't differentiate between eggs of both species. But you can use PCR and sometimes acid fast staining. So in the PCR, this method can provide a species-specific diagnosis when proglottid material is taken from the stool. This method requires specific facilities, equipments, and trained staff to run the test. This method has not yet been tested in controlled feed trials. So most common is stool tapeworm antigen detection, that is by using ELISA. Increases the sensitivity of diagnosis. The downside of this tool is at a high cost because kit is very costly. And in case of serum antibody test, that is by using immunoblot and ELISA. Tapeworm specified circulating antibodies have been detected by this technique. Human primary hosting is best diagnosed by microscopy of eggs in feces. So that is the most common and cheaper method for diagnosis, not only in this parasite, that in case of all parasites you can use stool staining. Now comes on the next part, that is treatment part, most important. So you can use praziquantel 5 to 10 mg per kg single dose, and also you can also use niclosamide 2 gram as a single dose, albendazole, that is a antiparasitic medicine. The treatment of cysticercosis must be carefully mentioned for inflammation reaction to the dying worms, especially if they have moved into the brain. In some cases, worm can be surgically removed, and in other cases, you can use these medications. A vaccine to prevent cysticercosis in pigs has been studied. The life cycle of the parasite can be terminated in their intermediate host, that are pigs, thereby preventing further human infection. The large scale use of these vaccines